Dear students, we will continue with the lipids. Now we will talk about compound or conjugated lipid. They contain additional group. Three different types of conjugated lipids we will study. Phospholipids, glycolipids, proteinolipid. They are also known as lipoproteins. Phospholipid, the lipid contain extra or additional group as phosphate and nitrogen group. In glycolipid, the additional group is carbohydrate. In proteinolipid, the additional group is protein. Now, if you look at the structure of phospholipid, again these phospholipids are of two different types. Okay, one is glycerophospholipid and another one is sphingophospholipid. So glycerophospholipid are the one which contain the glycerol as alcohol. So first we'll be talking about glycerolipid here. So this is again the glycerol structure, esterification of fatty acid. You can see at position 1 and 2, R1, R2 are two different fatty acid, and at 3 you can see this phosphate group is attached, and along with the phosphate the nitrogen group. Now this is represented by X. X could be a different nitrogen group. It could be, if suppose X is choline, then the phospholipid which is formed will be phosphatidyl choline. So choline chemical structure is this. So phosphatidyl choline is formed. This is also known as lecithin. If the X group over here, the nitrogen group is ethanolamine, then the phospholipid is known as phosphatidyl ethanolamine. Phosphatidyl ethanolamine is also known as cephalin. Similarly, phosphatidyl serine, phosphatidyl inositol. Now, if you see the role of lecithin, it is used as emulsifier, and it also plays role in cell membrane formation. Cephalin plays role in blood coagulation. Phosphatidyl serine plays role in cell signaling. Phosphatidyl inositol and serine both plays role in cell signaling. Now, if you see here, sphingophospholipids, another category of phospholipid which do not contain alcohol, glycerol as alcohol, but they contain sphingosine as a alcohol. One of such lipid is sphingomyelin, which plays role in signal transduction. Now, glycolipid, if you see their role, they constitute white matter of brain and myelin sheath of nerve fiber. Lipoproteins occur in cuticle of plant and also in the suberin of cork cell. So, occurrence and their significance. Now, we'll talk about derived lipid. They are also known as precursor lipid. They are produced during lipid metabolism and they do not contain fatty acid and glycerol. So they cannot be hydrolyzed. So just like other fat which contain glycerol, when you hydrolyze them, they will be forming glycerol and fatty acid. But since these do not contain glycerol, so there is no hydrolysis which is possible in their case. We'll discuss some because they are little complex lipid. Okay, the structure is complex, so we are not going to discuss right now the structure in detail, but we'll just see what kind of lipids are there, such as sterols, terpenes, eicosanoids. Sterols such as cholesterol, ergosterol, terpenes such as carotenoid, eicosanoids such as prostaglandin, leukotrienes, thromboxanes. Now let's talk about the sterol. Sterol are known as steroid alcohol as they contain 3 cyclohexane ring and 1 cyclopentane ring. So this is what is the structure of sterol ring. Okay, so in plant they are called phytosterol, in animal they are called zoosterol. For example, ergosterol, stigmasterol are type of phytosterols, and ergosterol is a provitamin form of vitamin D2 which is known as ergocalciferol. So the active form of vitamin D is calciferol, but the vitamin D calciferol is synthesized from ergocalciferol, and ergocalciferol comes from the ergosterol. So this has to be essentially consumed in our body for the synthesis of vitamin D. As you know, vitamin D is synthesized in presence of sunlight. So the sunlight, in presence of sunlight, ergocalciferol undergoes certain transformation and produces active form of vitamin D. So this is how our body synthesizes vitamin D from its precursor. Now this ergosterol also occur in the cell wall of fungi. So when we are doing the mushroom cultivation, after the harvest, the mushrooms are irradiated to the sunlight to increase their vitamin D content, or artificially irradiated to increase their vitamin D content. Here in animal we have cholesterol. Now cholesterol plays very important role in the body. It helps in the synthesis of bile. You know, bile is a emulsifier. It helps in digestion of vitamin A, D, K, E. It protects the internal organ, and it synthesizes important hormones such as testosterone, progesterone, estrogen. All these are steroid hormone. Okay. Now let's talk about the eicosanoids. They are the group of locally acting hormone derived predominantly from arachidonic acid. So prostaglandins, they are extremely potent biological substance that are produced by almost all the cells in response to cell membrane injury. They also play important role in glomerular filtration, and they also act as a vasodilator. Leukotrienes, they are produced by leukocyte by oxidation of arachidonic acid, and they are released in response to allergy, and they facilitate mucus formation. Thromboxanes act as a vasoconstrictor. They are released by platelet and facilitate platelet aggregation. So these are the eicosanoid which come under the classification of derived lipid, and they play important role in our body. Now overall, if we gather the function of lipid, we have seen individually also, but overall if we gather the function of lipid, then main function is energy source. Like 1 gram of fat on oxidation provide around 9 calories of energy. Okay, so good energy source. Then they act as a food reserve and storage reserve, like in the seed, in the plant. In case of plant, in the seed, fat reserve provide nourishment to the growing embryo, whereas in animal they get deposited as subcutaneous adipose layer under our skin. Then the same subcutaneous fat act as an insulator and provide insulation against heat. Then they also act as a shock absorber, and they found you can see the fat layer around the visceral organ where act as a cushion and absorb the mechanical shocks. They act as a solvent. They also act as a structural component of cell membrane. They also help in the synthesis of hormones. Now there are few other properties you can see, we'll discuss. They are important, like amphipathic nature of lipid. Okay, now if you see all the lipid, the waxes, fat and cholesterol, they are all hydrophobic in nature, although they contain the polar and non-polar group, but overall nature of these molecules, they are hydrophobic. But if you see phospholipid, they are having polar and non-polar both the part, but they are amphipathic. Okay, so an amphipathic molecule is a molecule that has both polar and non-polar part, and polarity develop in them. So that's why they are amphipathic. The phospholipid are amphipathic. Now let's see its two pole. One is fatty acid, another one is phosphate group, which forms the head. Fatty acid forms the tail. It is hydrophobic in nature, whereas phosphate, it is hydrophilic in nature. So that's why the polarity developed. So amphipathic molecules are biologically useful because they can interact with both polar and non-polar substance. Due to amphipathic nature, they form lipid layer in which the polar head faces outside, whereas the two fatty acid chain faces other chain of another phospholipid molecule in cell membrane. Now you can see in the cell membrane they form a bilipid layer. So the head is there and tail, two fatty acids are there which forms the tail. So two different phospholipid molecules, they are arranged end to end by their tails, and this tail region form the hydrophobic interior. Okay, so this is how they are arranged. Now phospholipid in aqueous solution um, forms micelles, in which the polar region faces outside, uh, and outside the surface of micelle, and the non-polar region forms the core. Now micelle formation, they are actually spherical in shape, and in which the um, polar and non-polar part are arranged like this. Toward interior is the non-polar part, and head is lying outside, which is in contact with water. Okay, now the micelle formation is different from chylomicron, which is formed during the digestion. Chylomicron is known as ultra low density lipoprotein that contain triglyceride, phospholipid, cholesterol, and proteins. They transport dietary lipid from the intestine to different part of the body. Okay, then you must have heard about liposome. Let's discuss. They are actually artificial vesicles. Okay, so man-made, which are spherical in shape, and whose membrane resemble that of phospholipid, that is bilipid layer. So here you can see the membrane is bilipid layer, and these are used as vehicle in the drug delivery. So you can load your drugs in this liposome, and then it is administered in the body. So this is the artificial formation of lipid which is used in the drug delivery. Now there are certain myths and fact about lipid. We'll just go through. Certain type of fat, especially the saturated and trans fat, increases the risk of cardiovascular diseases, diabetes, cancer, and obesity. Triglycerides store unused calories and provide energy. Cholesterol travel in the blood in combination with protein called lipoprotein. There are two different types of lipoprotein, low density and high density lipoprotein. Low density lipoprotein is called bad cholesterol, whereas high density called good cholesterol. The bad cholesterol can deposit on the walls of artery and narrow the arteries and increases the risk of heart diseases. That's why it is called bad cholesterol. Whereas good cholesterol, it carry the low density lipoprotein away from the arteries and back to the liver, where LDL is broken down and passed from the body. So high density lipoprotein is good. Now you can convert low density to high density lipoprotein in your body by following the healthy lifestyle, that is by consuming fibrous food, by consuming good, you can say, fat, and by doing the exercise. All these activities do this conversion. So good fat is present in olive oil, sunflower oil, seafood, canola oil, soya oil. So this is all about the lipid.